Hello everyone, this is Medical Gateway and today we're going to study endocytosis. So before studying endocytosis, let's discuss that what are the basic mechanisms by which any ion or molecule can move into the cell. So there are two methods. Number one is diffusion and the other one is active transport. So active transport is against the concentration gradient by the use of energy, and diffusion is from an area of higher concentration gradient towards the area of lower concentration gradient. So in diffusion, if the substance is lipid soluble, then the substance will simply pass through the lipid bilayer very easily. And if the substance is not lipid soluble or the substance is water soluble, then that substance has to pass through the pores. Okay, so this is diffusion and the active transport. Now, other than the diffusion and the active transport, there's also one other mode of transport, which is endocytosis. Now, what exactly happens in endocytosis is, for instance, these are the molecules which have to enter the cell, and this is the cell. Now, these purple colored molecules, when they want to enter the cell, first of all, they will come and they will be attached to these green colored receptors on the cell membrane. When these molecules are attached to the receptors on the cell membrane, there will be depolarization of this cell membrane. And this depolarization of the cell membrane will cause the movement of calcium ions in the cell. And when these calcium ions move in the cell, then what will happen next is that inside the cell there are contractile proteins. So these are the contractile proteins inside the cell. So there are three types of proteins. One is clathrin and the other one is actin and myosin. As you all know, actin and myosin are the famous contractile proteins, but you also have to remember clathrin here in this case of endocytosis. Now, on the movement of calcium ions inside the cell, these contractile proteins, the clathrin, the actin, the myosin, they all will contract. And when they all will contract, what will happen? This area of the cell membrane will form an invagination, and these two borders of these pit will move towards upwards and they will join each other. And when these 2 points will join each other, they will join each other and they will fuse. And when they will fuse, this will cause the movement of the whole pet along with its receptor and the molecules inside the cell in the form of an endocytotic vesicle. Okay, so this is the whole mechanism of endocytosis. First, the molecules are going to attach to the receptors, then there will be depolarization and movement of calcium ions in. When the calcium ions are moved in, the contractile proteins will contract and the pet will start to move inside and their edges will fuse, forming the endocytotic vesicle. That's how these molecules are going to move inside the cell by endocytosis. Okay, now, so these are the steps. First, there is attachment, then depolarization, then calcium moves in, then the proteins contract, then pet invaginates, the edges of the invaginates, they are fused, and which will result in the formation of the vesicle. Now, the endocytosis is of two types. One is the pinocytosis, and the other one is the phagocytosis. Now, what is the difference between pinocytosis and phagocytosis? The ingestion of minute particles is pinocytosis, whereas the ingestion of large particles is phagocytosis. So minute particles ingestion is pinocytosis and large particles ingestion is phagocytosis. Now, pinocytosis takes place in most of the cells of the body. Most of the cells of the body have the ability to do pinocytosis, whereas only few of the specialized cells can do phagocytosis, like macrophages, the WBCs. Okay, so this is endocytosis, pinocytosis, and phagocytosis. Now, what is going to happen with this vesicle when it is being endocytosed? Now come to this diagram. For instance, these are the particles which are going to be endocytosed inside the cell, and this is the endocytotic vesicle or pinocytotic or phagocytotic vesicle. Now what is the fate of this vesicle? Now we have to see that what basically is the significance of these particles. There might be some food particles, like there might be some lipids, proteins, or carbs, or there might be some foreign invaders which are being endocytosed. Now when this endocytotic vesicle is formed, the lysosomes inside the cells will come and join with these endocytotic vesicles, and when the lysosomes and this endocytotic vesicles, these both join, this will result into the formation of this digestive vesicle. Now this digestive vesicle has those particles which were being endocytosed, as well as this digestive vesicle has enzymes present in the lysosomes. Now the enzymes of the lysosomes will definitely break down these particles. There might be some food particles, there might be some foreign invaders, and when they will digest those particles, there will be residual body left behind. Now what is the fate of this residual body? This residual body will then be excreted out of the cell by exocytosis. The whole story started with endocytosis, then lysosomes are attached with the vesicle, endocytotic vesicle, then digestion takes place in digestive vesicle, and then the residual body left behind is exocytosed or excreted out. Okay, so this is the whole story. So endocytosis first, then endocytotic vesicle is added to lysosomes are added to endocytotic vesicle, forming the digestive vesicle, and then the residual body is exocytosed. Okay, so this is the endocytosis. Now we're going to study another concept, very important, which is autolysis and autophagy. Now what is auto? Auto means self, and lysis is breakdown, and phagy means eating. So autolysis is self breakdown, and whereas autophagy means self eating. Now what is the difference between these two terms? Autolysis means that whenever a cell is damaged, now what happens? The lysosomes inside the cell are ruptured. And when the lysosomes are ruptured, their whole enzymes come out of the lysosomes. And these enzymes will start eating the digested portion, the damaged portion of the cell. And if the damaged portion is in the larger quantity, these enzymes have the ability to digest whole of the cell, the entire cell. Okay, so this is known as self-lysis or autolysis. Whenever a cell is damaged, the lysosomes rupture and their enzymes come out of the lysosomes, out of their barrier, and these enzymes start eating the cells. So this is known as self-eating or self-lysis or autolysis. Now what is autophagy? So autolysis is a pretty uncontrolled phenomena because it starts with the damage to the cell. Now what is autophagy? Now autophagy as compared to autolysis, autophagy is a pretty controlled thing. It is self-eating. For instance, now any organelle which is worn out, okay, the lysosomes take that organelle and break it down, not causing lysis of the complete cell, just the killing of the damaged or worn out organelle. So this is known as autophagy. Now this it isn't like that the whole lysosome is ruptured and all the contents of the lysosomes are coming out into the cytoplasm. The lysosome eats the organelle. Okay, and when the lysosome eats the organelle, it results into the formation of autophagosome. Okay, as previously discussed, when the lysosomes are attached with the endocytotic vesicle, it results into the formation of digestive vesicle. Exactly like that, whenever an organelle is eaten up by lysosomes, this will result into a formation of autophagosome, not releasing completely all its enzymes outside into the cytoplasm. So all of the cell is safe from the lysosomal enzymes, just the worn out organelle is being digested by the lysosomal enzymes. So this was autophagosome, and this whole process is autophagy, which is to eat one self. Okay, so this was endocytosis, pinocytosis, phagocytosis, autolysis, and autophagy. See you in next part.